My name is Jackie Lees, and it's a pleasure to welcome you back to the afternoon session, where we're really focusing now on talks from former members of the Koch Institute, all of which actually who are really great examples of convergence. So the first speaker is Giovanni Traverso, and he's actually an example of combining science and engineering and medicine. He received his BA from Trinity College at the University of Cambridge in the UK, and then his PhD from the lab of Burke Vogelstein, where he focused on developing novel tests for early detection in colon cancer. He then went to medical school at the University of Cambridge and came to the States to conduct his internal medicine residency at the Brigham and Women's Hospital, a gastroenterology fellowship at the Massachusetts General Hospital, and postdoctoral research in the Koch Institute with Bob Langer. In that position, he actually transitioned now from biology into the field of chemical and biomedical engineering and developed novel technologies for drug delivery, as well as physiological sensing via the gastrointestinal tract. So he recently started his independent research career as the Carl Van Tassel Career Development Professor in the Department of Mechanical Engineering here at MIT. He's also a gastroenterologist at the Brigham and Women's Hospital, and he's continuing to work both on diagnostic tests for early cancer detection and devices for cancer monitoring and drug delivery. And the title of his talk is Transforming the Oral Route, New Approaches for Delivering Macromolecules, Creating Ultra-Long-Acting Therapies, and more. So please welcome Gio.
Thank you so much, Jackie, and thanks to Matt, Tyler, and Jane for the opportunity to share some of our work. And it's really a pleasure to stand here and share some of this work that actually started during the transition when the Institute started. So I was there, and I participated in actually moving into the building over 10 years ago now. And what I've done here is selected two areas, or really vignettes, and I should say they're journeys because they continue, but started at that time, over a decade ago. And so just before I dive in, a lot of the work that we do, we are really passionate about really trying to make an impact, and we either start companies or work with companies to try and bring those technologies to patients, and these are my disclosures. Now, as far as, what is it that we do? I think Jackie sort of captured what we do, but I include this picture. These are my three kids, just two plus years ago, really next to a GI tract from the Bodies exhibit at the Science Museum. And the reason that I include this is not only to share with you my wonderful three children, but really also to give you a sense on the scale, or the scale that we're working in. And I think it's important to think about length scales. I know that many of my colleagues that spoke earlier and are speaking after me often focus on the molecular side of things, and I think from an engineering perspective, it's important to recognize the challenges, but also the opportunities in working in this environment. And so hopefully it gives you a sense. And I guess from a scale bar perspective, my children at the time were 3, 6, and 8, so hopefully that gives you a little bit of a sense on the size of the intestines that we're looking at in the stomach. And really what we're trying to do through our work is address a fundamental challenge, and that is that although we have many therapies, many of those therapies are never taken or taken inappropriately. And actually, when one looks across disease, what we find is that around 50% or so of folks actually fail to take medication as prescribed. And over the years, some people have said, well, it doesn't happen in cancer, it doesn't happen in X. And specifically to oncology, given the current audience, what I wanted to do was just, and I apologize, let me just show you this graph right here. And this is actually taken from a review from Ann Partridge's team over at the Farber that really examined the adherence to oral therapies. And it's just to say that, this is a problem that's germane really across medicine. And what we're trying to do here is develop technologies to help address one of these major challenges. And so as engineers, what we did was really focus on a fundamental observation. And the observation that we focused on was recognizing that patients, individuals, are more likely to continue to take a medication when it is taken more infrequently. And so I can I've experienced this with antibiotics, for example, over the course of my lifetime, where I would much rather take a drug once a day than 3 times a day. And certainly many trials have actually demonstrated this. This is actually taken from a review from the New England Journal highlighting this correlation as far as infrequency being associated with higher adherence rates. What's really interesting is that over the past decade or so, there have been a few other trials looking at a very specific class of medications, and these are bisphosphonates. Bisphosphonates are drugs that are typically used for the treatment of osteoporosis. It's one of the very few classes of drugs that exist in formulations that go beyond the 24-hour period, so weekly, monthly, and actually even yearly. And this particular trial out of Japan, but there's similar trials out of Europe as well as North America, demonstrates and highlights the continuum that we observe in patients, that as we stretch out those intervals, it appears that individuals are more likely to stick with their therapy. And so really recognizing this fundamental observation, we set out to do the following. And this is a cartoon from The Economist from a few years ago when we were starting on this journey that I feel really captures what it is that we're trying to do, and that is develop an orally administered metered dosing system, hopefully in a smaller form factor than what you see here, of course. And now, this is a great vision, but there's some fundamental challenges, of course, in front of us. And the main challenge is one that we experience daily, which is that material transits through our bodies, typically in the order of a day or so. And so as we start to think about how we overcome this physiologic process and consider, for example, a standard capsule, that capsule will be ingested, transit. Typically, the majority of drugs are absorbed in the small intestine, and if there are any remnants of that dosage form, they will be excreted. Many groups and companies have actually developed systems to start to slow down and increase the opportunity for drug release. And they've done that, for example, through the incorporation of mucoadhesives. And certainly that will give one, a few more hours, essentially, of bioavailability or exposure. But really what we wanted to do was have a system that could reside in the body, not for a few hours, but rather days, weeks, or potentially even months following a single ingestion event. And so what I'd like to do is just walk you at a high level some of the challenges that we recognized early on. And the first challenge is one that I typically refer to as the ship in the bottle problem, and that if we want to have something that changes shape in the stomach, we need to traverse the esophagus. So somehow we need to get that ship through that esophagus and then, of course, open up again. And in order to do that, we have an encapsulation challenge. We have to somehow fit this ship inside of a capsule. So that's challenge number one. The second challenge is that ship or this dosage form needs to unravel, unfurl, and then reach the gastric cavity. Now, upon unfurling or unraveling, we need these systems to then reside. And we need them to reside in a way that they're not causing any blockage, but also maintaining the drug stable in a fairly harsh environment that experiences pH variation between 1 to 5 or so, as well as, of course, being exposed to the broad range of the human diet. And then finally, after being retained, we need that system to break up and then be excreted safely. And so what you're seeing here in these photographs, X-rays, are essentially the first, the earliest demonstrations of the residency, and that you can see that on the X-ray on the left of the screen. And on the bottom right, there are some remnants that are radiopaque, and those are those remnants falling apart and passing through the GI tract of a pig. Now, generally, when we tackle a challenge, one of the first things that we tend to think of is what could go wrong. And one of the things that we were concerned early on was having these systems transit through the pylorus, through the exit of the stomach, into the small intestine and open up there. And so in that setting, we put our heads together and developed a new material. It's a material that we refer to as an enteric elastomer. The term enteric refers to the capacity or behavior of that material to remain stable in an acidic environment, in this case, what you see labeled as SGF, or simulated gastric fluid, and unstable in a near neutral environment, in this case, SIF, or simulated intestinal fluid. And so what you're seeing there on the graph is essentially the mass that's remaining with 100% at the top and 0 at the bottom, with the x-axis reflecting time. So that reflects the enteric nature of the material, and then the elastomeric nature is what you see shown on the right. That we needed a material that was stretchy, that we could fold and incorporate into some of the early prototypes. And that's exactly what this material allowed us to do. We took that material, we interfaced it with other materials that have been used for drug delivery. In this case, we use a material called polycaprolactone, and we showed that we could create these different structures. And early on, we made these circular structures, showed that we could place them in capsules, and then upon placement, let's say in simulated gastric fluid, the capsule would dissolve, but those intervening segments of enteric elastomer would remain stable. And if placed in a near neutral environment, they would actually start to break apart. And so this was all well and good, but we wanted to now test some of these concepts in different designs to see if these systems would open up, unravel, and remain stable. And so these are some of the early X-rays demonstrating those early opening events inside of the stomach of a pig. But just to give you a little bit more up close and personal view, this is actually a very early video of one of these star-like systems overlying the pylorus of a pig. Just want to highlight the tissue here looks healthy. There are no abrasions. Also, the gastric cavity is empty, and this is on an overnight fast, reassuring us that these systems would not block essentially the transit of material, but also that they would not induce any damage in the mucosa. So we were reassured on the engineering side as far as sort of being able to develop an encapsulated system that could reside in the gastric cavity. But at the time we were working with the Gates Foundation, and one of the big areas that they're interested in tackling is malaria. And what I have here are representations of the mosquitoes that carry the parasite. And there had been an observation that had been made in the 2000s, which had to do with a drug called ivermectin. Ivermectin is a drug that's used for the treatment of a range of parasites, and there had been a specific observation that actually ivermectin was also toxic to the mosquitoes that carried malaria. And so the foundation team asked, could you help us develop a system that could provide a sustained level of ivermectin, which is already being used in the field in mass drug administration campaigns. And so we said, we think we can. And actually, this is actually the first 3 experiments we did in pigs, demonstrating that we could maintain a level above the toxic threshold to the mosquito, which you see there in the red dotted lines. And you can see the comparator in the bottom of the slide of the approved medication. By the time we actually published this the work, we actually had demonstrated that we could maintain those levels out to 14 days. And I should say that just a week ago, the first human was dosed with these dosage forms, so it's now in clinical trials. Now, building off this work, we recognize the modularity of these systems, and so we recognize that we could actually put different drugs or different materials or formulations to provide different pharmacokinetics or release patterns of different drugs. And so the next challenge that we tackled was actually HIV. And we looked at all of the drugs at the time that were available. We looked at the ones that were potent enough, essentially, such that a week's worth could fit inside of a capsule, and identified these 3 and developed formulations with a whole range of different materials, and shown that we could, with a single capsule, provide coverage of the antiretroviral for 1 week. Now, the last thing that we did academically was a once-a-month oral contraceptive. So we extended this work beyond the 1 and 2 week period, really out to a month. And in this case, we loaded the capsule, the Starlight capsule, with levonorgestrel, and demonstrated that we could maintain the levels of levonorgestrel for greater than 21 days. And so with this, we actually transitioned the work into a company. The company is called Lindra, which we co-founded now a few years ago. And I just want to share, this is data from a recent phase 2 trial. And one of the things that when we started on this journey that we considered was the capacity of these systems to provide the sustained release, but also to have some potential additional advantages. And one of those being that by providing a continuous steady release, we may be able to lower the peaks that are, for example, associated with daily administration. And for many drugs, those peaks can be associated with toxicity. And this is data directly in humans showing comparable levels between daily administration of a drug. In this case, it's risperidone, which is to be used for schizophrenia, but also that lowering of the Cmax of that peak now when the drug is administered using this dosage form. Now, let me switch gears to the second vignette, and this is one that has been an incredible journey starting back in 2009, 2010, and I've had the incredible pleasure of working with several folks over the years. I include their photos here. Carl Scharlhammer was a graduate student in Chemie. I was on his committee. Alex Abramson was another graduate student in course 10 who I co-advised. Esther Caffrell was a postdoctoral scientist that I co-advised. And Ulrich Rabeck has been an incredible collaborator at Novo Nordisk. And what I'd like to do is just share with you two clinical cases. And these are two patients that I looked after when I was a GI fellow back at the MGH. But just before I get there, really at the heart of what we're trying to do here is address this fundamental preference that patients, but also healthcare providers have for the oral route. So in general, the majority of folks actually prefer to take a pill over an injection. But as we know, many drugs cannot be administered orally and need to be administered through parenteral means. And so recognizing this and really inspired by the next two cases that I'd like to just sort of dive into before we get into the technology side. And these are two cases that I'll never forget. And the first one, as often happens when one is on call, I was on call on a Friday, and I was called to the emergency department to see a gentleman who had ingested shards of glass. And it was a witness event. And I walked down to the emergency department, and I saw this gentleman sitting there with a half-eaten sandwich in his hand. And so I approached him and I said, sir, please stop eating. I'm the gastroenterologist on call. And then I informed the emergency department team there wasn't much from an interventional perspective that I would be able to do. And I reviewed this case with with the attending who was on call and explained the situation. And he said, he asked me what was the size of the shards of glass. And so I went back, I reviewed the imaging, and actually there were a couple of shards of glass, and they were just around 2 centimeters or under that. And what he suggested was, we can observe this gentleman over the weekend. And he said, I suspect he'll be fine. And this to me was just mind-blowing, in that to think that actually we would just observe this patient who had ingested shards of glass at Mass General was just sort of astounding, but was extremely instructive. And this person did absolutely fine. He was discharged on Monday, and actually the shards of glass were radiopaque, so we were following them by X-ray. And so we could observe when they passed, and the patient was none the wiser insofar as when they passed. But what it really taught me and gave me a much deeper appreciation for how the GI tract can tolerate sharp objects. Now, the second case is one that's a little bit more common that we tend to see, I would say, relatively frequently. And this is a lady in her 60s who's been taking a lot of ibuprofen because she's been having quite a bit of joint pain as well as back pain at the time. And she comes into the emergency department complaining of epigastric pain, which is in sort of the upper part of the abdomen overlying the stomach. She also describes her stools as being black and tarry and feeling lightheaded. And so this sort of constellation of symptoms and signs generally makes us think, potentially that there's some bleeding. And additionally, if someone's been taking a lot of ibuprofen, which by the way is a wonderful drug, but also has the side effect of inducing ulcers, we certainly worry about potential active bleeding. And lo and behold, in this case, we performed an endoscopy, and there was an actively bleeding ulcer in the stomach. One of the things that we do as gastroenterologists is we inject that ulcer with epinephrine, with adrenaline. And the reason that we do that is for a local effect, a local vasoconstrictive effect, because what we want to do is really get a good look so that we can then intervene appropriately. But as soon as we give that injection, which is for a local effect, what we recognize is that within a few seconds, all the alarms in the room start bing-bonging for the very simple reason that whatever we injected for the local effect also has a systemic effect on the heart. So now putting these together, we started to wonder, could we develop systems that could be ingested that potentially might be able to give micro-injections of a fairly broad range of molecules? And so at the time, with some minimal resources, we put some ideas together. We did some basic prototyping in the machine shop, not too far away from here, and fed these to pigs. We were able to get 3 pigs. We fed them these capsules. The pigs were fine. There was no damage, no abrasions to the GI tract. And then the other thing that we did was, my wife is an endocrinologist, and I asked her, I said, could you help me get some insulin? And so she was kind enough to get me some insulin samples. And what we did was we injected insulin in the GI tract, in different parts of the GI tract, and we looked at the kinetics of the hypoglycemic response, so how quickly the glucose levels would lower. And what we observed was actually that the kinetics were either comparable or even better than, for example, subcutaneous administration. So this was really reassuring and actually resulted in a tremendous amount of interest from industry. And our colleagues at Novo Nordisk really moved very quickly with a major collaboration, which has been really terrific. But when we published that initial sort of early, I should say, concept paper, there were three major challenges that we had in front of us. Number one was, if we wanted to develop a system that was ingestible, that could give or provide a microinjection, somehow we needed that system to allow or enable approximation of the business end or the injection site with the mucosa every single time. Number two, we would need to develop a system that would autonomously trigger to actually give an injection. And then number three, we needed a way of administering a sufficient amount of drug. And what we wanted was a device that was the size of a blueberry, approximately, such that we would minimize the risk of obstruction. And so there are some boundary conditions around how big of an object, a solid object, can reliably transit through the GI tract. And it's a little bit around a blueberry, a little bit bigger than that or so. And so let me walk you through how we tackled this. So we took a page from the leopard tortoise as well as weeble wobbles and developed an approach where we use capsules that can self-orient. And that was how we addressed challenge number one. The way that we developed this, we did quite a bit of fundamental modeling to identify the optimal shape as well as density distribution. We tested this with high-speed photography in different media, but also in pigs in simulated walking and active movement. And what we observed was that actually these systems would always self-orient. And so we're reassured that using this approach could reliably provide active and good engagement with the mucosa. Now, for the second challenge, which is that autonomous actuation, we took a step back and sort of asked ourselves, are there some universal cues that we could hone in on and then use and apply to materials? And the cue that we honed in on was humidity, that we recognized that being in the stomach, the environment is one that has very high humidity, and if that's not the case, then the individual has bigger problems. And so using that cue, we then started to think of materials that would be sensitive to the humidity. And we needed a material that could essentially maintain a stored energy and then release that stored energy all of a sudden. So what we wanted was a material that displays brittle fracture mechanics. So I want you to think candy cane, not taffy. And actually what we used was exactly that. We used sugar glass. And so what you're seeing here is one of the very early prototypes of a miniature spring that is coated in sugar glass, which when placed in a humid environment can release that stored energy all of a sudden. So we have a potential solution for number one and two. And now for the third challenge, we have sort of this device that we want to keep in a form factor of approximately the dimensions of a blueberry. And so when thinking about the drugs, let's say insulin or potentially later monoclonal antibodies, et cetera, many of those drugs are really are administered in liquid format. And liquid formats really are generally limited by the solubility of the drug in the solvent. And so what we wondered was, could we just remove the solvent? Could we make a solid dosage form? And that's exactly what we did. So we created very small needles that were made of up to 100% insulin. And what we did was that we showed that we could administer those, and the levels that we would administer using the system were comparable to the levels achieved when administered subcutaneously. So this was really, the first description of a way of administering a biologic using a self-orienting, autonomously actuating system. And so, but in front of us, we still had a couple of challenges that we really were interested in tackling. We recognized that if we could deliver fluids, that could open up some new avenues. And that has to do with both the type of drugs, but also the kinetics of absorption. And so what we did was actually we completely redesigned the system and developed a system that has a needle that can actually provide an injection and then retract that needle. And it can, when it's engaging with the mucosa, it administers a small liquid bolus, and we optimize the system so that the distance of penetration is consistent every time, providing a essentially submucosal depot of that liquid. Now, just to give you a little bit of up close and personal, I just have a very brief animation. Let me just show it right here of how it works.
A timed dissolving actuator prevents the pill from delivering the payload too early or too late in the digestion process. After delivery, the insertion mechanism retracts back into the device and the pill passes safely through the body.
So using this liquid self-orienting system, we actually showed that we could deliver many other drugs. So we delivered adalimumab or Humira, insulin again, a GLP-1 analog, and then also epinephrine. Now, with the system and recognizing that we're still in the middle of a pandemic, we also wondered if we could deliver other types of molecules. And the most recent work that was published was actually the delivery of mRNA. And so using this system really coupled also to some fundamental material science to generate formulations of the mRNA that could directly interface and optimally interface with the gastric mucosa, we demonstrated that we could, using the system, deliver mRNA in pigs. And so here is that data, and this just came out a couple of months ago. And finally, this technology was just dosed to the first subjects just over 6 weeks ago, approximately. So we're thrilled that at least these systems are moving into humans. So with that, I have the tremendous honor and pleasure of presenting here the work of many. I just want to introduce you, at least at a high level, literally, of some of those team members. This is back in 2016, to many of the people that contributed to the early work on the STAR system. In 2018, to many of the members that contributed to the self-orienting system, the first generation, as well as most recently last summer, to folks who've contributed to some of the material science and the second and, third generation now of the self-orienting system. I also want to thank Bob Langer, who's been an incredible mentor, colleague, and friend over the years. And then with that, if I don't get to your questions, please feel free to email me, and I have my emails right here. So thank you so much again for the invitation. It's really a pleasure to be here.